So I'm going to talk a little bit about using liquid biopsies for personalized medicine. And I want to set the stage first, talk a little bit about the change that we're seeing in medicine today, where we're moving away from using morphological diagnosis, just looking at a slide. We're actually moving into more molecular diagnostics, where we're looking at tumor mutations and RNA profiles, etc. And for that, you need a tissue sample, or you used to use a tissue sample, and sometimes you can't get a representative sample out of the tumor. And we say the tissue is an issue when you want to do molecular profiling. And now with this liquid biopsy concept, you can actually get access to the same molecular markers without the need for a tissue sample. And that is helping both the diagnostic field move forward and the personalized medicine field move forward, but also the therapeutic field is moving forward faster, because this type of liquid biopsy enables you to stratify patients better and do faster enrollment into clinical trials, which can lead to drugs reaching the market quicker. When you're thinking about a tumor, for example, they are often heterogeneous, and they're heterogeneous both in sort of space and time, because they can change very quickly. So we know that a tumor can actually change the dominant molecular signature in as little as 3 to 4 months after initiation of treatment. So having a liquid biopsy sample enables you to monitor that over time. And in addition to that, it enables everyone to be their own baseline, which is a unique feature of these biomarkers, where it's very hard to find a biomarker that is identical across the entire globe, because we're all different, and different people have different levels of different biomarkers. But for you to validate that one, you have to find something that doesn't change too much between individuals. The liquid biopsy space enables you to be your own baseline, where you can take a sample at baseline where we can all be different, but then you monitor how that changes over time, which is very helpful. The liquid biopsy space is dominated by 3 different biological entities. So we talk about circulating tumor DNA, we talk about exosomes, and circulating tumor cells. What's important to understand when we talk about these types of liquid biopsies are that they are very different molecular entities, and they enable you to profile different things. Circulating tumor DNA is released from the tumor cell when it dies. Basically, it's the dying process of the tumor when the cell goes through necrosis or apoptosis. Versus the exosome release is an active process. It's a metabolically active process that happens in the tumor. So that's the living part of the tumor. The circulating tumor cells are seen after the tumor have evolved and gotten this dissemination process started. So usually in the later setting of the disease process. When you look at, so this image here to the left illustrates 2 tumor cells growing in vivo. And what's fascinating here is that you see almost this cloud of vesicles being released from these tumor cells. And we know that over 20,000 of these vesicles can be released from a single cell in a day. And they package a lot of material, RNA and proteins and metabolome and other things that can be used for biomarkers. But tumors don't release these exosomes to be used as biomarkers. They use it, there's actually a purpose behind them. And exosomes are known to be part of the disease process, and that's very important because that makes it more relevant as a use as a biomarker. And we know, for example, that tumor cells release exosomes to stimulate tumor cell growth. They block the immune response on every level, both the innate and the adaptive immune response is actually blunted by molecules found on exosomes from tumors. And they can also stimulate angiogenesis. So they're loaded with biomarkers like RNA and proteins and DNA. There's a lot of confusion about what is actually packaged inside these microvesicles, and there's a lot of research going on in the microRNA field. However, we know that the entire RNA transcriptome can be found inside these vesicles. So we can find the mRNA, the microRNA, and the long non-coding RNA. And since RNA is really the language of the cell, nothing will happen inside a cell without it being reflected on the RNA one way or the other. You can use this to really profile and understand what's going on inside a cell at any given time. And exosomes aren't limited to tumor biomarkers. So we know that exosomes are released from all types of cells. And in the case of neurodegenerative diseases, we see exosomes can be loaded with tau protein, alpha-synuclein protein, and even prion proteins. So they can be used for biomarkers in that field as well. So I like to illustrate this tumor immunity cycle as an example of exosomes being part of all these molecular processes that happen. So when a tumor is growing, you have antigen release, and in the tumor immunity cycle, but that we're trying to sort of master now with drugs. So there's a lot of things happening in the immuno-oncology field, where we're trying to make sure that the antigen presentation and activation of the immune response happens inside these tumor patients. And in that context, PD-L1 is an important molecule, and PD-L1 as a checkpoint inhibitor is used today as a biomarker in tumor tissue. However, what I want to illustrate here is that in all these processes that happen in the immunity cycle, exosomes are released in every step of the process. So exosomes are released from the tumor, as you can see antigens in these exosomes. You have exosomes being released at the antigen presentation stage, the priming and activation, cell trafficking, etc. So every part of the tumor immunity cycle is represented by exosomes. And that's been recognized now that exosomes play a role. And we can actually see that exosomal PD-L1 is increased in tumors, in plasma from tumor patients. And we also know that the exosomes carrying these molecules are actually part of the disease process and promote the tumor growth in vivo. So when you're studying exosomes, of course, the first step of studying exosomes is to take out the biofluid sample and then select a way to isolate your exosome from that biofluid sample. And there's a lot of different ways of doing that. There's probably over 100 different methods of exosome isolations. However, how you do it is very, very important. And you have to optimize your processes depending on what you're trying to achieve with the biomarker, what type of biomarker you're looking at. These are examples of platforms that we currently use for clinical applications. So we have the ExoRNA that's used for research, but also the Exolution platforms that are used for clinical exosome isolation. And different types of kits are used depending on what the purpose is, what type of biomarker you're looking at. And it's critical to select the right way of isolation because every method there are different pros and cons with different methods. And you have to understand what type of biomarker you're looking at. So the best method for exosomal protein analysis may not work for exosomal RNA analysis and vice versa. And you have to understand your biomarker also, if it's uniquely found in the exosomes or if it's a ratio of extravesicular material versus intravesicular material. And if you know that ratio, you can figure out how pure your sample needs to be before you process your biomarker. And I want to highlight that there are cGMP versions of these exosome isolations that actually produce extremely robust data. So this is an example of exosomal RNA extractions over 4 different operators and 2 different days. And you can see that the replicates are basically identical. So this is taking a plasma sample, and you isolate the exosomes from the plasma sample, crack them open, get the RNA out, do the reverse transcription, and then do qPCR of it. All those processes have the same sort of standard deviation as you expect from just the PCR alone. So it's basically a very robust process. So we know that the exosome isolation step is not a hurdle that you can't overcome. Another very important factor of exosomes is that they carry with them antigens from the cell that released the exosome. So you can utilize this biology to your advantage, because when you're looking at a plasma sample or any biofluid sample for that matter, they're really complex. They're full of exosomes and other vesicles from a variety of processes, and most of the exosomes are typically not from the target that you're actually interested in. And the exosome biology can be utilized to then enrich your sample of choice. So you pull down with positive selection the exosomes of interest, or you use negative selection to remove noise in the system. If you know that, for example, the majority of the exosomes are coming from red blood cells and platelets, and those are not your target organs, then you can remove them and do a negative selection. So by utilizing these exosomal markers that are present on the surface of these vesicles, you can enable brain-specific or tissue-specific biomarker analyses. And you can also enable earlier cancer detection using your liquid biopsies. If you isolate your exosomes correctly, you can do a lot of different things with the exosomal RNA. For example, we've optimized processes for every type of biofluid, so plasma and serum, CSF, urine, etc. And isolating the exosomal RNA, if you do it correctly, you get the entire transcriptome out. This is where there's a lot of controversy in the literature, because when you do your isolation, if you do not do it properly, you will degrade your RNA, or partially degrade your RNA, and you will only see small RNAs. So if you do it properly, you can actually get the same diversity of RNAs and the same type of detection of RNAs as you see from a tissue sample. So from 0.5 ml of plasma, we see the same diversity and breadth of RNAs as you would see if you take a tissue sample and sequence that. So you see over 30,000 out of the 56,000 genes, or you see over 17,000 of the 20,000 mRNA that are in gen code. And in every sample that you run, we typically spike in 92 different exogenous RNAs as a control at different levels, so that we know that the sequencing is quantitative and what the limit of detection is in every sample. And as you can imagine, sort of that when you can profile the entire transcriptome, it opens up a lot of opportunities for using these exosomes in biomarker research. Here's an example that I like to show, because this focuses on early stage cancer detection, which has been sort of the holy grail of liquid biopsies and is really challenging to achieve. Most of the other liquid biopsy platforms are you can use them in the later stage setting, but it's very challenging when you're trying to go into early stage detection. In this case, it was stage 1 and stage 2 ER positive, HER2 negative breast cancer patients. Took 1 milliliter of plasma from patients and controls, and these weren't patients and normals. They were actually women that had a scan and had a suspicious scan, and the biopsy either showed early stage cancer, or the nodule that was seen was benign. So in that setting, we actually had access to the FFPE material as well. And when we sequenced the tissue from the tumor and the benign lesions, Of course, the benign lesions look different than the early stage cancers. That was expected. However, when we took total plasma exosomes and sequenced them, we actually saw almost a similar distribution of patients and control, that we could actually separate the patients from the early stage cancers. But the signatures were not identical. So the signature in the plasma exosomal RNA wasn't what we saw in the FFP material, and that is because we utilized total exosomes. And most of the exosomes are coming from other processes than the tumor. So it comes from the immune response and tumor stroma, etc. So what we did in this case was that we took another aliquot of plasma from these patients and pulled down with positive selection breast cancer specific exosomes. And with that method, so you pull down the specific vesicles, you get the signal out of the noise. So you actually start to see what's coming truly from the tumor. And when we did that, we were a little bit surprised to see the nice correlation that we achieved. Among the patients that overexpressed this marker that we use for positive selection, the plasma sample clustered next to the tissue from the same patient. So not only could we differentiate the patients from the controls, but we could actually tell you from the plasma exactly what the RNA signature looked like in the tissue. And that is sort of the holy grail of liquid biopsy, because then you can actually go in and define what processes are happening inside the tumor at any given time, but in a liquid sample. So there are different applications of these exosomes, and over the last decade we've worked on many different applications at Exosome Diagnostics. So you can use them to monitor treatment response, for example, in clinical trials, and help pharma companies stratify what patients are more likely to respond to their drug. You can monitor immunotherapy response. You can look at kidney transplant rejection or bladder cancer signatures. So basically anything on the planet and beyond, because you can also look at what happens in astronauts in space. So I mentioned we can actually use exosomes to pull down brain-specific signatures. And in this particular case, NASA wanted to understand if the astronauts that suffer from this syndrome when they go up into microgravity, called VIP, that gives them mental fogginess and some vision impairment. And in this case, we wanted to see if we can actually monitor some of the processes that happen in the brain of individuals with high intracranial pressure, which happens when you go up into microgravity. And we can actually do that. So you don't need access to a brain sample from these astronauts. So these are typically done by RNA profiling. Liquid biopsy today often focus on mutation detection, and you can do that as well. However, for mutation detection, I mentioned most liquid biopsies that are looking for mutations, they struggle to go into early detection. It's easier to find these mutations in later stage disease. And that's typically because in early stage disease, you don't have enough copies of the mutations in circulation. And since we have access to the exosomal RNA and cell-free DNA, and mutations exist on both, we figured why not combine the two? Because if you combine exosomal RNA with the cell-free DNA, you get a double whammy. You basically solve one of the biological problems, that you get more copies of your mutation, and you start to look at two different biological processes at once. So you're looking at the dying process of the tumor and the living part of the tumor, and increase your sensitivity of your mutation detection at the same time. So over the years, we've shown that this seemed to be beneficial in every case. So irrespective of what cancer you're looking at, you always benefit by combining the mutations on exosomal RNA plus the cell-free DNA. And you can use this for for non-small cell lung cancer, melanoma, colorectal cancer, ovarian cancers, etc. And we always see this added value. And to illustrate sort of some of that added value, when you're looking at cell-free DNA and that you get from a specific cell-free DNA extraction platform, and you compare the copy numbers of cell-free DNA that you get from a platform that combines cell-free DNA and exosomal RNA, you actually get the same copy number of the targets on the cell-free DNA when you add the exosomal RNA. So basically, trying to do two things doesn't impair your ability to isolate the cell-free DNA. When you add the reverse transcriptase into the mix and you actually combine the RNA with the cell-free DNA, you can see that in every case, these dots are now above the diagonal. So in every case, you get more copies of the target when you combine the RNA with the cell-free DNA compared to just the cell-free DNA extraction platform. And the amount of added value that you get depends on the patient. So we often get this question, how much added value do you get from the exosomal RNA? And it depends. So this particular patient up here, for example, have 16,000 copies of BRAF on RNA plus DNA, but only 3,000 copies on DNA alone. So it was the majority of the signal is actually coming from the exosomal RNA. Whereas the patients that are closer to the diagonal, they have their signatures dominated by the cell-free DNA fraction. But combining both of these actually makes it easier and for you to monitor things that happen over time, especially when patient goes on to treatment. Because when you have a patient on treatment, the copy numbers of mutations often go down significantly below the detection limit. But in this case, you can actually monitor the mutation load both in stable disease as progressive disease. On average, we see up to 10 times more copies of the mutation when you look at RNA plus cell-free DNA compared to just cell-free DNA. So this is an interesting study that was actually done blinded, where half of the sample was sent to Exosome Diagnostics for the combined approach, and half of the sample was sent to a cell-free DNA company that only looked at the cell-free DNA. And on average, we got 10 times more copies of the mutation, but it also increased the clinical sensitivity of your patients. In particular, in the patients that are known to be very difficult to find the mutations in. So the non-small cell lung cancer patients with intrathoracic disease, the M0, M1A types of tumors, they often secrete very low amounts of cell-free DNA. So it's usually below detection limit. So the clinical sensitivity with the cell-free DNA was 26%. However, when you add the exosomal RNA component to that, it goes up to 74%. So you get more added value when you have a patient that is very tricky to look at. And that makes sense because some patients have thousands of copies of the mutation on cell-free DNA. And then if you get 5,000 copies versus 1,000, it's the same result. It's positive, right? So you don't get the added value in those. This is another example of basically the same thing, where instead of doing a sequencing-based approach, we did a PCR-based approach. There's an FDA-approved test for liquid biopsy looking at T790M mutations. It's the Roche Cobas assay. So this one utilizes cell-free DNA. It's been published to have around 58% clinical sensitivity at an 80% specificity. We ran a study where we had a similar PCR-based assay, but that was able to pick up mutations on both RNA and DNA, and the clinical sensitivity went up to 92% at an 89% specificity. So we basically think that it's a wasted opportunity whenever you're looking at cell-free DNA alone. because you're throwing away half of your material, or actually 90% of your material in some cases.